Welcome back, everyone. This is the last episode in the immunology series, and today we're going to be focusing on the gut and autoimmunity. When talking about the immune system, and especially autoimmunity, it's inevitable to have conversations about the gut and the microbiome. The microbiome refers to a vast number of microorganisms that live inside the body. The microbiome consists of bacteria, fungi, and parasites that mainly reside in the gut, skin, and mucosal linings. In the gut, a single layer of enterocytes separates the intestinal lumen from surrounding tissues. This is often referred to as the epithelium. A protective mucosal barrier is formed by goblet cells who secrete mucus and Paneth cells who secrete antimicrobial peptides. This mucosa is also full of IgA, intraepithelial lymphocytes, and B cells. To drive home the importance of the microbiome, it is estimated that there are around 38 trillion bacterial cells in the body, and this outnumbers human cells by roughly 8 trillion. So in some ways, we are actually more bacteria than we are human. So it makes sense that the microbiome, including bacteria, plays such a large role in the immune system. Real quickly, we'll recap how T cells originate from hematopoietic stem cells located in the bone marrow. After maturing in the thymus, they become activated in secondary lymphoid tissues, including lymph nodes, the spleen, the sinuses, and the mucosa-associated lymphoid tissues known as the MALT, and a subcomponent of MALT called the gut-associated lymphoid tissues, also known as GALT. The gut-associated lymphoid tissue, or GALT, is important because it includes lymphoid follicles called Peyer's patches. Peyer's patches are made up of specialized epithelium that contain microfold cells, also known as M cells. These follicles are home to mainly B cells, with some T cells and dendritic cells. Because Peyer's patches form in the gut, they are exposed to a high degree of antigens from bacteria, fungi, viruses, and protozoa. As a result, these germinal centers in Peyer's patches are constantly active. While these germinal centers produce a wide array of antibodies in response to infections, they also generate large amounts of IgA to promote tolerance to friendly and commensal microorganisms. Pattern recognition receptors are host receptors that recognize molecular structures that are essential for pathogen survival. However, PRRs also recognize ligands from commensal bacteria, and this crosstalk is critical for maintaining tolerance between the host and commensal or friendly bacteria. PRRs differ from major histocompatibility complexes because they recognize a broad variety of ligands, where MHCs are specific to only one antigen. Although PRRs are innate in nature, both PRRs and MHCs participate in the crosstalk necessary for activating the adaptive response. Here are the three major types of patterns that PRRs recognize. We have pathogen-associated molecular patterns that are molecular motifs present on both pathogenic and non-pathogenic bacteria and viruses. We also have microbial-associated molecular patterns that are motifs on microbes and are essential for their survival. DAMPs are damage-associated molecular patterns, and these are released as a result of cellular stress or tissue injury by the host, and it helps to facilitate repair in response to damaged cells. However, improper targeting of DAMPs by PRRs can result in autoimmunity. Toll-like receptors are a type of pattern recognition receptor, and they're predominantly found on antigen-presenting cells, but they also show up on other cells like epithelial cells, neutrophils, and T cells. TLRs can be expressed on the cell surface or intracellularly on endosomes. TLR signaling enhances the expression of co-stimulatory molecules on APCs and promotes the release of pro-inflammatory cytokines. Here are just a few of the TLR functions. They help to maintain the integrity of the epithelial barrier by inducing secretion of antimicrobial peptides and mucins. They also stimulate the differentiation of T cells, epithelial cells, and germinal centers. They tighten intracellular junctions between epithelial cells. They also maintain tolerance to commensal bacteria, and they help to induce the release of hormones involved in peristalsis. We know that there are at least 10 or 11 different human TLRs that recognize different types of pathogens. Activation of TLRs can result in signaling cascades that aid in the defense of pathogens and repair damaged tissues. However, overstimulation can result in an excessive pro-inflammatory response and contribute to autoimmune diseases. For example, ligands like lipopolysaccharides, or LPS, are present in the cell walls of all gram-negative bacteria, and they're known for triggering sepsis. However, overstimulation of TLR4 by LPS can disrupt the entire microbiota ecosystem and result in leaky tight junctions and autoreactive B cells and T cells. Here are some of the diseases involved in TLR signaling, including rheumatoid arthritis, systemic lupus erythematosus, multiple sclerosis, and type 1 diabetes. So dysbiosis occurs in the presence of metabolic alterations, defective epithelia, and pathogenic microbes. These disruptions support the overgrowth of harmful microorganisms and are accompanied by a marked loss of diversity, and the overall abundance of the microbiota is diminished. Disruptions in this equilibrium between the microbiota and the host immune system is detrimental for epithelial repair and resolution of inflammation. It can also lead to a condition called leaky gut, where the tight junctions between the epithelial cells are compromised. The loss of barrier integrity allows undigested food particles, toxins, and pathogenic bacteria to leak across the gut into the bloodstream and provoke autoimmune responses throughout the body. While TLRs play a critical role in maintaining the integrity of the intestinal barrier, they're also implicated in the development of leaky gut. For example, TLR1 and TLR2 are responsible for tightening the junctions between the epithelial cells. However, TLR4 activation results in the loss of integrity, contributing to leaky gut. Medications, and particularly antibiotics, have been under scrutiny for their associations with dysbiosis and leaky gut. Infections also play a huge role, along with dietary changes and psychological and physical stress. Immune tolerance refers to the positive and negative selection of T cells and B cells. The negative selection against B cells and T cells is important so that the immune system doesn't mistakenly attack its own tissues. Central tolerance refers to primary lymphoid organs, including the bone marrow and the thymus. Peripheral tolerance refers to the bloodstream and secondary lymphoid tissues, such as the spleen, lymph nodes, and the GALT and MALT. B cells, for example, first undergo negative selection in the bone marrow. If a B cell receptor shows high affinity for a self-antigen, the B cell either undergoes editing of the receptor, apoptosis, or anergy. If an autoreactive B cell somehow escapes this negative selection in the bone marrow, they undergo further negative selection in the periphery, like in the lymph nodes, where they are again subject to either apoptosis or anergy. T cells similarly undergo negative selection in the thymus. However, deletion of autoreactive T cells in the thymus is only 70% effective. So T cells play a large role in the loss of central tolerance and a large role in the loss of peripheral tolerance due to their ability to activate and mediate B cells. One of the proposed mechanisms for the loss of tolerance involves the T cell receptor. So autoreactive T cells can have either a single or double T cell receptor with different configurations. When 30% of them on average escape the thymus, they can be activated by APCs that are presenting either self or foreign antigens. Activation of autoreactive T cells results in tissue damage through the increase of pro-inflammatory cytokines and cytotoxicity. Autoreactive T cells can also activate and mediate the B cell response, and that leads to an increased production of autoantibodies and tissue damage. In summary, loss of tolerance is when the immune system is no longer able to discern between the self and foreign antigens. Next, we'll review the three main mechanisms behind the loss of tolerance, including molecular mimicry, epitope spreading, including viral persistence, and bystander activation. Molecular mimicry is one of the main hypotheses explaining the pathology of autoimmune disease. It refers to a situation where an amino acid sequence or a structure found in self-antigens is similar to a foreign molecule or pathogen. The part that is recognized by the MHC or TCR is referred to as an epitope. When the T cell binds to self-antigens and to antigens from a foreign pathogen, the immune response can cross-react against its own tissues. However, there is a 99.7% overlap between human and microbial proteomes, so molecular mimicry alone cannot explain the pathology of autoimmune disease. Rather, it points to a combination of factors inherent to the host genetics and environment. Epitope spreading is an immune response that can target epitopes on the same protein or other proteins on the same tissue. Epitope spreading contributes to autoimmunity through 1. Persistent viral infection, 2. Continuous damage of self-tissues that cause the release of new self-antigens, 3. APC ingestion and presentation of the new self-antigens, and 4. Nonspecific activation of more autoreactive T cells. Bystander activation usually occurs with CD8 T cells. However, CD4 T cells are also being researched for playing a similar role. To begin with, we have a CD8 T cell that is activated in response to a primary antigen. Clonal expansion ensues during the acute stages of infection. During the resolution stage, memory CD8 T cells are formed that are specific to the first antigen. Bystander activation occurs when there is an exposure to a new antigen, and dendritic cells begin producing a lot of inflammation. As a consequence, these memory CD8 T cells that are specific to the first antigen can now become activated from the pro-inflammatory environment as well as stimulation by TLR agonists. Unlike mimicry, this bystander activation is not dependent on T cell receptors, and it is not specific to an antigen. There is growing evidence that bystander activation plays a serious role in cancer and infections associated with autoimmune diseases. There's still a lot we have to learn from the immune system, but one thing we do know is that the immune system is intended to protect the host, and sometimes it does that at any cost. We can see that with bystander activation, because the immune system is thinking, I have all of these memory CD8 T cells from this primary infection that I could repurpose to use in this new infection, and that of course helps to kill infected cells. However, the downside is that when these nonspecific CD8 T cells are activated, they can also accidentally attack healthy tissues from the host in a type of friendly fire. And this type of overreactive immune response is what contributes to irreversible autoimmune diseases. Well, that's it for this immunology series. If you liked what you learned and want to support this channel, you're welcome to follow me here, or you can find me on patreon.com, where there will be other opportunities for learning and engagement. If you stay posted, my next episode will be covering the saturated fat controversy and who is right, saturated versus unsaturated, and we're going to break down all of the nuances in between. Thank you for joining me on this journey, and I look forward to seeing you on the next one.